Okay, so I'm seeing our numbers are stabilizing, so I'll go ahead and get going. We'd like to welcome you today to MSU Extension's Vaccine Program webinar on immunology. My name is Carrie Schreier. I'm an extension educator with MSU Extension's Children and Youth Institute, primarily focusing on parenting education. And today we're excited to welcome MSU Professor Dr. Kathy Hoag. Dr. Hoag is an immunologist with MSU's Biomedical Laboratory Diagnostics program, and she's joining us today to talk more about immunity, how vaccines create immunity, and how immunity works in our bodies. I'll let Dr. Hoag introduce herself, and then we'll go ahead and get going.
Thank you, Carrie, and thank you for inviting me to be the guest today. Good afternoon, everyone. I am a native of Michigan, so I grew up in the far western end of the Upper Peninsula of Michigan. My dad was a copper miner, and I went on to do my bachelor's degree at Michigan Technological University up in Houghton. So I stayed in the UP for quite a while. But then I really ventured off and I went to Texas and got my graduate degree there at the University of Texas Southwestern Medical Center at Dallas. And then I ended up here getting an appointment at MSU in 2001. So I've been teaching here at MSU for a little over 20 years. Hard to imagine I've been here that long, but I really enjoy what I do. And I'm looking forward to helping you understand how your immune system works and how the COVID vaccine can really help us fight this current ongoing pandemic.
Great. Thank you so much. That's a beautiful part of the UP. I love it up there. We're glad to have you. So just a reminder, we are taking live questions today. You're welcome to go ahead and type your questions into the Q&A, and we'll add them to the queue of questions that we're working from. We will take questions from Facebook as well. You're welcome to message the My Stronger Family page with your questions, and we'll be sure to make sure that we add those in. Additionally, we will be sharing in the chat a link to a demographic form. We appreciate your taking the time to let us know where you're coming from when you join us today. This is an anonymous link, but it just helps us track where we're reaching people across the state of Michigan. So with that, I'm going to go ahead and jump in with the first question that we've had submitted. And this is one I think many of us have heard a lot about, but it still gets a little confusing. So the first question is that really basic. How does COVID-19 infect us?
Sure. So COVID-19 is really very much like a lot of the other respiratory infections that we have. It's initially taken up just simply by breathing it in from really tiny droplets in the air around us. And then the virus very specifically uses a protein on the surface of our own cells lining our respiratory tract to dock to those cell surfaces, and then it injects in its nucleotides, basically the recipe for making more viruses, and it takes over the cell that it infects, so that now that cell produces many, many more virus particles, which we then breathe out and pass on to others. From the respiratory tract, that virus can also spread to other parts of the body. Of course, you swallow virus accidentally. You don't do it on purpose, but you're going to. So if it's capable of infecting gastrointestinal cells, part of your intestine, which COVID is, it can also infect those cells and cause symptoms of gastritis in individuals as well.
Great. Thank you. Our first question here from our live audience is, if you have a strong immune response to the vaccine, does that mean that you have a strong immune system?
In general, sure. I would say so. And we do know that there is variability from individual to individual in our population and how well they respond. There are many things that we know influence how well you may respond to a vaccine or a natural infection. We know that older individuals, their immune system becomes quite a bit less effective at handling infections, in particular virus infections. And so that's why we see older individuals being encouraged to make sure they get the flu vaccine every year, or they get a vaccine against shingles, which is a reactivation infection of chickenpox, which they could have been exposed to as children. So we know that the elderly have less effective immune responses. But there are also within our human population a lot of variable genes that you inherit from your parents that impact how well you respond to particular organisms or not. So, as a population, that variability protects our population as a whole, but sometimes individuals are not protected as well.
Great. That's so interesting. It's been really interesting to observe the different ways that people have responded as we've kind of moved through this pandemic and the vaccines. My apologies. All right. So the next question we have here is, does immunity from a COVID infection last longer than immunity from the COVID-19 vaccine?
That's actually a very good question. I don't think there's evidence to say that that's true. I think the immunity that you get from the vaccine is pretty equivalent to the immunity that you get from a natural infection. And in fact, we've seen reported that people who have had COVID infection in the past can get it not only once, but 2 or 3 more times as the new variants emerge in the population. I think what is also similar between the vaccine and natural infection is that we certainly do know that once you're exposed in some way, whether it is the vaccine or the natural infection, that your likelihood of severe infection, hospitalization, and possible death are greatly diminished. So the benefit of being exposed at least once is important. And that's really probably based upon the fact that our immune system retains memory. So once it's seen a particular infectious agent, it goes back to those memory responses, and it's much faster in adapting to another exposure to that same organism.
So on that subject of vaccines and the new variants, could you talk a little bit about how or if the new boosters are different from the old boosters?
Sure. So I'm going to confine this mostly to the mRNA vaccines, which are the ones that the FDA recently approved. And they're what you could consider bivalent boosters. So they contain everything that the initial mRNA vaccines produced by Pfizer and Moderna had. But they have the added mRNA sequence of the Omicron variant in there. Overall, they differ very, very little. So the way that they manufacture the virus, the way that they package it and store it and distribute it to the pharmacies, the way that it's administered and the dose administered is absolutely unchanged. The only thing that has been added is the new, molecular signature of the virus as it's evolved within our population.
All right. Well, and as a follow-up to that, we were actually just discussing this before the webinar began. Are you as likely to have a reaction to the booster as you were like to the original vaccines, or the people who are avoiding those big reactions or want to schedule their vaccine for a Friday so they can be sick on Saturday or something like that?
Yeah, I'm actually in that boat myself. I'm looking at scheduling my booster still, and I'm trying to figure out around my travel schedule and teaching schedule and everything. It appears that if you responded to the initial vaccine, you're going to respond to each subsequent vaccine booster about the same. The reason is that vaccines have to be designed to include an element that your immune system is going to detect as dangerous, right? In order for the immune system to respond, it has to get that danger signal initially. And if it's exposed to a new protein or a new particle it's never seen before, and there's a lack of a danger signal, your immune system is going to ignore it. So the fact that you do respond to the vaccine is actually a good sign that your immune system is activated and it's going to provide you with beneficial protection against that infection.
I'll tell you, I thought I was sick for a day there following my booster, but I was much sicker when I had COVID. So I'll take that one any day. On that note, we did have somebody share in the comments, they got their new booster last week and then had to take the next day off sick. So maybe that mental note to schedule that in. Much easier to schedule than a surprise COVID infection.
Yeah, and I think probably the longer you have between your previous booster or if you've had COVID, the longer time between the two, the more aggressive your immune response is going to be. Because if you're within the last 3 months or so, you still have antibody remaining, protective antibody that's going to neutralize some of that vaccine proteins, and it's not going to be as effective for you. But if it's been a while beforehand, then it's going to respond much more aggressively.
All right. So the next question coming up here, and this is kind of a little bit more nuanced, more of a bit of an opinion here. How would you respond to someone that is not vaccinated and says that they had COVID-19 maybe one or more times, so they're not worried about needing to get vaccinated anymore now that they've had COVID?
Well, the one thing I guess I would really focus on is that the vaccination programs that we have, not only in the United States, but worldwide, are really designed to protect the whole population, right? And as we discussed earlier in this talk, not everybody can mount a helpful immune response to a vaccine. The elderly, those that we know who are highly susceptible to COVID-19, such as the obese, those with cardiovascular disease or lung issues such as chronic obstructive pulmonary disease or asthma. They're all much more sensitive and susceptible to severe infection. So the benefit maybe of you getting a booster is not so much to protect you, but to protect your loved ones and those around you in the population who are more susceptible and their immune system simply is not capable of protecting them. So if we get as many people in the population vaccinated as possible, it won't allow the virus to circulate as easily in the population, and it'll protect those people who are more susceptible. That's really the goal of vaccine programs that we have here in the US.
Right. Yeah. To keep the people who are more vulnerable from getting ill.
Yes.
Comparison, I heard that, and that I can move on to another question, but I found this to be very interesting that with rubella and the original vaccinations for rubella, that program was really put in place, my understanding is, to help protect pregnant women.
Oh, absolutely. Yeah, rubella is one of a couple of different viruses that can cause very severe birth defects in young, or even sometimes lead to stillbirth if the mother acquires a initial infection with rubella during the second trimester pregnancy. So it's one of those that physicians should definitely screen young women for immunity against rubella and caution them to, use birth control or some other pregnancy prevention method until they can get vaccinated and protected against rubella.
That's so interesting. I as a parent, it's not until this point in my life that I learned so much about how some of these vaccines are really in place to help us protect other people
Right.
in our world. Great. So the next question here is about the difference between the COVID vaccine and other vaccines that we've had. So the question reads, I've heard a lot about how different the COVID vaccine is compared to other vaccines. Can you talk a little bit about the difference between mRNA vaccines and traditional vaccines?
Sure. And this is actually a fascinating question, and one that I'm really excited to talk about, because when I first heard that the US was developing the mRNA vaccines, I was really impressed. I mean, I really think that this is groundbreaking new technology in vaccine development that we honestly have had a lack of for decades, right? We haven't made many new breakthrough kind of increases in technology in vaccines for quite some time. So vaccines have really existed for a very long time, right? It was actually the very first one documented in medical literature was produced by Edward Jenner in 1796, so quite a while ago, right? And what he did is he made an observation that milkmaids seem to be especially protected against infection against smallpox, which smallpox at the time was ravaging populations. And those people who got infected, 25% of them died from the primary infection. It was really a deadly virus. And he looked into the fact that milkmaids were protected. And what he discovered is that cows had a very similar virus infection to smallpox called cowpox. And that when the milkmaids milked the cows, they were exposed to the cowpox, and it was similar enough of a virus that it protected them from the smallpox. So he took basically lesion fluid from cowpox and used it to vaccinate people who had never been infected with smallpox before and show that it could provide protection. And so that was really the very first vaccine that was ever used. And then it went on for, decades and decades after that, where we would use either whole, very closely related viruses for a vaccine against viruses, or killed bacteria to produce a vaccine against particular types of deadly bacterial infections. But there were dangers in the design of those vaccines. A couple of the most prominent ones is that the organisms could mutate during the production process and become more pathogenic than what we want them to be and actually cause disease instead of protection. And we actually saw that happen with one of the polio vaccines that was produced. And polio is a virus that can mutate, and that happened. So there's one type of polio vaccine that's just not used anymore because of that. The second danger of using these, slightly modified but live organisms or even killed organisms is that people who have or hidden or previously undiagnosed mutations in the way that their immune system functions can get a really aggressive infection, even from a vaccine, right? And we can actually see this in the human population. So our vaccine adverse reaction database that the US CDC keeps documents those kinds of really adverse infections in response to vaccine. And most of the time, they can actually be tracked back to an unknown gene defect in the person who got the infection.
Wow, that's so interesting. So much I didn't know about the technology and everything that goes
Yeah.
behind
So
it.
these new mRNA vaccines, then the benefit of that is that we're not giving the whole organism. We're giving a very defined tiny piece that cannot actually ever cause an infection. And so that really eliminates the dangers of the first kinds of infections or first kinds of vaccines that we've had, and some of the side effects that those could have.
Well, on that sort of note about how these vaccines work, one of the comments that our next question leads right into is that they've heard that the COVID vaccine can alter people's DNA, or that it's working to alter their DNA. Is that true?
No, I can give an emphatic no to that one. So we have what we call in biomedical sciences a central dogma of molecular biology, which means we inherit DNA in the nucleus of our cell from our parents, right? When the little embryo forms, we get some DNA from dad, some from mom, and together they make us. That DNA is basically the template for how our whole body is going to be created. The DNA gets what we call transcribed or copied into messenger RNA copies. And then that messenger RNA is translated into the functional proteins. Our body doesn't go backwards. We don't go from protein back to RNA, back to DNA. And so the mRNA within the vaccines that we have that were produced by Pfizer and by Moderna cannot go backwards from mRNA into our DNA. We just don't have the enzymes within our human cells to do that. Now, there are actually some viruses that do this, that are very dangerous viruses and make it very, very difficult for our immune system to fight them. And the poster child for that is HIV. So HIV is called a retrovirus, retro meaning backwards, right? Because it does have its own enzyme that it inserts into our cells that causes it to go into a DNA form, and then it splices that DNA into our own DNA. But that's one of the very few viruses that can do that. The
Wow.
messenger RNA vaccines, though, do not provide any mechanism for that to happen.
I've heard this mRNA vaccine sort of referred to as a Trojan horse, that it's kind of slipping that fight in there.
Yeah, you could think of it that way. So it's the virus itself, right? As we said early on in this talk, it docks to the cell and then it injects in its nucleic acids. What this mRNA vaccine is doing is actually delivering that nucleic acid signature, that RNA, mRNA, but in the little lipid dropule. So it just uses a little lipid carrier, right? It's basically, you could think it's shaped like a ball, right? And then inside the ball is packaged all the mRNA. When the ball hits our cell surface, that it kind of explodes and the mRNA is delivered right into the cell.
That's so interesting. All right, well, we had a question come in here from Facebook about boosters. And the question is, if I've already received a 4th booster, should I additionally receive the new Omicron booster?
Yes, I'm in that same boat. I've gotten 4 of the original, well, I had a mix actually of Pfizer and Moderna, which is what they recommend, but I've had 4. And yes, I will be getting the Omicron. I'm trying to schedule it next week. So the new vaccine contains new proteins that the Omicron variant makes, and they're significantly different enough from the original virus, which was used to create the original vaccine, that it takes a while for our immune system to catch up. And that's how we can get a true infection again with Omicron, even though we've been protected with the previous vaccine. So by getting this new vaccine, you're giving your immune system a head start, right, to protect you against these new variants that have arisen.
Okay, great. So we've learned so far, get that vaccine, but you might want to schedule it on a day when you can take a day off work if you had a reaction before. All right, perfect. And on that trend of who should get vaccines and when we need to get them, the next question is, my OBGYN is recommending that I receive the COVID vaccine. I'm assuming person is pregnant. What would be the advantages to getting the vaccine now while I'm pregnant versus waiting until I've had the baby?
There are actually a couple that I can think of. Number one, we know that the vaccine induces what we call antibody immunity. So what antibody is, it's actually a soluble protein that's produced by a particular cell in your immune system called a B lymphocyte. So when the B lymphocyte encounters the vaccine proteins, it gets activated, and then it secretes, or it produces from the cell, a protein that can seek out that pathogen. So it can float around through all of our body fluids and in our blood. And wherever the virus is located, it can bind to the virus. It can prevent it from infecting new cells by just basically coating its surface. It can also mark it so that other cells in the immune system then can recognize it as foreign and can destroy it. So we know there are 2 different ways that mothers pass on immunity to their babies from infections that they have been exposed to in the past. One is during the 3rd trimester, the antibody in the mom's bloodstream is actively transferred across the placenta from the mom's bloodstream into the baby's bloodstream, giving the baby protection against every single infection that mom has experienced in her whole entire life. And so that antibody after birth lasts about 3 to 6 months and protects that baby from infections when their immune system is still very much developing and they're really pretty susceptible otherwise. So that's one way. The other way is that there's another kind of antibody that's produced not just in the bloodstream, but in mucosal surfaces, so your respiratory tract, your intestine, called an IgA type of antibody. And IgA is highly concentrated in breast milk. And so when a mother breastfeeds her baby, they continue to receive protective antibody, which basically protects the barrier surfaces of the upper respiratory tract and the gastrointestinal tract against very commonly acquired infections. So moms do a lot to protect their babies even after they're born.
That's so interesting. I knew that there was a connection there, but I never knew kind of how that worked. The next question here is about cytokine storms. I'm not sure if I'm pronouncing that.
Cytokine, actually.
Cytokine. Great.
Yeah.
Thank you. So the question is, it's my understanding that most of the damage done by the COVID-19 virus occurs when the body's immune response releases a cytokine storm. What is a cytokine storm and why is our immune system doing this?
Well, first, I think I have to stop and define what a cytokine is first, right? So a cytokine is just simply a protein. It's a small protein produced by one cell to communicate with other cells. And the immune cells are probably one of the most potent producers of cytokines. So when you, for example, get a new infection and you get a fever or you feel really tired and sore, that's all caused by cytokines being released from your immune cells, causing different changes in other body cells in response to the infection. So, for example, if a cytokine called tumor necrosis factor is released during an immune response, it causes the chills and the fever that you experience. Well, bacteria and viruses are designed, they're basically selected to replicate the best at regular body temperature, right? At 98 degrees. And if you raise that body temperature, they don't replicate as quickly, right? So a fever is actually a good sign that your immune system is responding and it's caused by your immune system. But if we look at COVID-19 infection in particular, one of its dangers is that it infects a lot of different body cells and a lot of different body tissues. So many viruses are very tissue specific, cell specific. COVID can kind of spread throughout the entire body. And when it does that, it alerts immune cells throughout the body. And when you get that many activated at the same time, they overproduce cytokines. And that's what we call the cytokine storm. So you could really think of it as too much of a good thing. You know, there's saying in the human population, everything in moderation, and your immune system response is no different. It follows that rule too. And so in a very severe infection, it can actually do damage by responding too aggressively. And that's essentially what a cytokine storm is.
Wow, that's so interesting. So basically your immune system is overreacting to the situation at hand, which
it's kind of overwhelmed and it's overreacting. It's kind of like frantic at that point.
right. It's that toddler having a temper tantrum on the floor instead of
A little bit
to correlate to my parenting stuff. All right. So this is the next question really is something that I observed in my own house when our whole family got COVID at the same time, and my kids ranged in age from 9 to 18 at the time. The question reads, COVID-19 infections seem to create different immune responses in children and adults. Why does age affect this immune response?
Well, just like emotions and brains, your immune system in young children is not fully developed yet. And so in particular, this relates to the T lymphocyte part of your adaptive immune response. So T lymphocytes come from a little organ that sits above your heart called the thymus. And the most active new production of T lymphocytes occurs all the way in young people up through puberty. And then the function of the thymus kind of wanes through the rest of your life. So you work really hard during those early years up until puberty to create the whole population of functional T cells that are going to sustain you and your life for the rest of your life. So it's still learning. It's still learning what are the organisms out there that are dangerous and that they have to respond to. It's building up the memory cells that will be able to respond to them. And it's still kind of immature in the way that it does it.
Okay. And so that affects how strong of a reaction your body.
Yeah, absolutely. If you have fewer cells that are capable of responding to an infection, then it's just not going to be as aggressive.
Wow. That's the answer to why my 9-year-old skipped right through things when I was down and out for the count.
There's also another aspect to it, which is kind of fascinating. Children's tissues are really sensitive, right? And so, and they're also, their immune system is not only learning what's dangerous, but it's also learning what's not dangerous. And it's important to have that balance. So young people are actually, their immune system is kind of programmed to be tolerant to things. And that's important because you don't want to respond aggressively to food. You don't want to respond aggressively to, things that you can inhale from the environment, like grass pollen and tree pollen, right? And when we see that, we see allergic reactions, we see food allergies, we see hay fever and all of those kinds of things. And in that case, your immune system is responding aggressively when they actually should be tolerant. And so what we see is that young people's immune system are actually kind of defaulting to that tolerance zone. Okay. And to dampen the immune response to be, so that it is less aggressive. So some of it's active and some of it's just, I haven't seen it before. And so it's still learning.
Wow, that's so interesting. So much I didn't know here. And I thought I'd been brushing up hard on preparing for this, but that's really interesting. Great. Okay. Well, our next question loops back to Omicron and the variants we keep hearing about. Why does the COVID-19 virus keep changing and turning into all these different variants?
The short answer to that is they're sloppy. So when they copy their they have actually not mRNA genome, they have a single-stranded RNA genome. It's slightly different, but pretty close. But the enzymes that the virus produces are sloppy. They put in the wrong matching nucleotide when they're copying it and making new virus nucleotides to go into the new virus particles. And they also have no way of correcting the errors when they make them. So if they make an error, it just gets packaged and it goes out in a new virus. If that error just randomly happens to provide that virus an ability to escape our immune system and to replicate faster, then that virus very quickly out produces all of the ones that don't have that mutation, and it expands, it gets passaged on to other humans, and there's our new variant. So viruses in particular are pretty sloppy, and they actually kind of, you could say, do that in a way that helps their own pathogenesis, because it allows them to escape our immune system with pretty regular, timing. And we've seen that, right? If we look, it's probably every 5 or 6 months, we get a new variant of concern that begins spreading around the globe from the initial place where it evolved out as people travel.
So mentioning, the place where the new variant evolved, we noticed at the beginning, it seemed like they were calling those different variants by the location where they were first identified, and now they're using another naming system. Can you tell us how the variants are named?
Yeah, so the World Health Organization is kind of responsible for naming the new variants. And, there was a lot of focus really in the beginning of COVID-19 that oh, China produced this variant or this virus, and they were responsible for the outbreak. Well, no, that's just where it arose. And so, since then, new variants have arisen in the UK, in Britain, all over the world, really. And so the World Health Organization says, no, we need to put a stop to the stigma of where these variants are coming from. And so they just simply chose Greek letters of the alphabet to name subsequent variants. And so this is really pretty common in biomedical sciences. For example, T cells, the particular receptor they have comes in 4 different varieties, and we call them alpha, beta, gamma, and delta. So we've seen those same COVID variants arise as COVID has continued to move through our human population.
Gotcha. That makes sense. All right. Well, we're going to switch tactics a little bit here. And I know in addition to your work directly with immunity and immunology, you also do some work around testing. Is that correct?
Mm-hmm. Yeah, that's right. So I'm a medical laboratory scientist certified, and that's what my initial education was. And now I teach students who are studying to become medical laboratory scientists.
Perfect. You're just the right person to answer these questions then. Is there a reason why you have to swab for 10 seconds? So I'm assuming maybe the nasal swab for 10 seconds.
Correct. Yeah. So when you do these antigen tests, right, the over-the-counter tests that you either could, order through the U.S. Postal Service or buy at your local pharmacy or even Meijer, these antigen tests, if they're going to be positive, always show 2 lines, right? There's a C line and there's a T line. The C line is a control line and the T line is the test. So it's whatever you're trying to detect with that protein assay. The C line is a built-in, what we call positive control. It tells us that the test is still working properly. All of the enzymes and different reagents within that test kit have not degraded, right? That they're still working. The C line is always detecting something that every human should have in a cell if they collect an adequate sample. So when you're rubbing that swab against the inside of your nose, you're picking up epithelial cells that line your nose. And then you're putting it into the solution in the little tube that comes with the kit. You're washing it off, and then there are enzymes that bust open those cells, and they're detecting protein from those epithelial cells in order to create that positive C line. If you don't get a C line, then either your specimen collection was inadequate, you didn't rub right, you didn't get any real sample there. So you're not going to get a positive T line whether you tried or not, right? Or you got an adequate sample, but there's something wrong with the kit. Either it was stored at too high a temperature, you bought the kit on the way to work, and then it sat in your car and it got heated up to 120 degrees while your car was in the sun all day, and all those enzymes degraded. And so the kit itself is no good. So it's basically just kind of a built-in control to make sure that you got an adequate sample and that the kit itself is working properly.
Okay, that's so interesting. On that subject of kits and whether or not kits are working well, the next question is about the expiration dates on COVID-19 tests. It says, I noticed that my COVID tests at home have expired. Why would home COVID tests expire?
Well, for many of the same reasons that I kind of mentioned in the previous answer. Storage of these test kits is not always ideal when they're released to the general population. People who don't have air conditioning in their home, the temperature could get too high during the day, and rapid temperature changes can degrade the reagents within the test kits. So over time, we also can see things degrade. The plastic containers that they're stored in can release things into the solution, into the reagents over time that can interfere with the way that they function. And so the FDA has basically said you have to put an expiration date on all test kits, just like we see with over-the-counter drugs, right? Those all have expiration dates. And usually it's around a year, sometimes a little bit longer. I think in the case of these over-the-counter test kits, most of them have been closer to 6 months, just because, we don't have that much experience with how stable these kits are because they were all kind of created so quickly, right, in response to the pandemic. And I think over time, when the companies producing them are able to look at how stable they are, some of those expiration dates might get a little bit longer if they can prove to the FDA that they're still functional later than what the current expiration date is.
Would it be, I mean, if you took a test kit at home and it tested positive, is it possible that it was expired and you could get a false positive because it was expired, or?
No, the concern is really a false negative. If you get a positive, then you can be really quite confident that it is truly positive. The risk is that it becomes less sensitive and you would get a negative test even though you truly are infected.
Okay.
That's why they also tell you that if that, control line doesn't show up, you need to retest and consider the test invalid.
Okay. And you're heading right into this next question that popped up in the chat. If the test kit is expired, but the control line is working, does that mean that the test is accurate?
Oh, that's kind of a wishy-washy one. As a laboratorian who's certified, I would tell you don't trust it if it's expired. But from decades of experience, I can tell you that if it says you're positive, I would trust it. If it says you're negative, I'd probably follow up with the PCR test that you can get done at a real hospital lab. These tests are going to lose the sensitivity for detection over time. And if they're past the expiration, I would be less inclined to trust a negative test result. Okay. Even though the control is positive. You'll see the control is going to get lighter and lighter further you are out from that. And so you're going to miss the real slight positives on those tests.
Sure. Speaking of those tests and storage and expiration, you mentioned that they can get too hot. Can they also get too cold?
Yeah. Proteins do odd things. When they're frozen, right, they can misfold. And most of the reagents that we use for these tests have a protein component, an enzyme that has to function in order to make the test more sensitive, so that we can actually see a result visually. And if you freeze them, the proteins can fall out of solution so that we lose their active function. You can also have them dehydrate, and then they don't dissolve well when they come back into suspension. So think about freezer burn. If you keep a steak in your freezer for too long, a similar thing can happen with these kinds of reagents if you freeze, thaw. So you really want to store them at a pretty stable room temperature as much as possible.
That's good advice heading into the winter in Michigan. Don't leave those test kits in your car when it's cold either.
It's like your pets, right? Yeah. Just like your pets. Don't leave your pets in a hot car. Don't leave your pets in a freezing car.
Good. Always good advice. All right. So on that, how sensitive test kits are, the next question is, I've heard that the current strain of COVID is taking longer to test positive on tests. Is that true?
For the over-the-counter protein antigen-based tests, I would say it is not as early, right? So, but the PCR-based test, I would not say so. They are still very, very sensitive for the new Omicron variant. We're not really sure what the basis of that is. There might be just enough protein changes that the tests, which were initially designed against the initial spike protein, are no longer as sensitive. and that's probably the basis of it. So we may see the development of new tests that are specific for Omicron. I anticipate we will.
Okay. I was just going to ask that follow-up question. I certainly had that experience. I knew I'd been exposed. My whole family was sick. I started to have symptoms, but I was a solid 3 or 4 days in before I tested positive.
Yeah. If you have symptoms, I would say at that point, go and get the PCR testing at a urgent care or ready care or whatever, local COVID testing sampling site you have available.
And speaking of PCR tests, are the antigen home tests as accurate as the hospital PCR tests? Sounds like you were just saying not really.
Well, I got to use a little bit of distinction in terminology. So accurate in laboratory lingo does not mean the same thing as sensitive in laboratory lingo. So if we look at accuracy, that means if you get a positive test result, the person really has the infection, right? And I would say the accuracy of the two is actually quite close. If you get a positive on a home test, you're going to get a positive on the PCR test, pretty much guaranteed 99.99% of the time. But the home tests are not as sensitive, which means you could get a negative test result at home, even though you do actually have an infection that could be picked up by the PCR test. So we have to be careful with our terminology. And laboratory medicine, we have more defined terms that we use. So I would say they're not as sensitive on the home test kits, but they certainly are as accurate.
Okay. So once you're really symptomatic, you're quite likely to be getting an accurate test at home with the nasal swabs once you're sick.
Yeah.
Okay. Good to know. I'm flipping back a bit to our vaccinations. The next question is, how do we know that the boosters are safe? They seem to be being continually created so quickly. How do we know they're safe?
Well, all of the human studies that we did prior to release of the first mRNA vaccines by Pfizer and Moderna are using the same production platform, the same lipid dropules to deliver the vaccine, the same chemicals, processes used in their production. They're packaged the same, they're shipped, stored, delivered, all exactly the same. The dosage hasn't even changed. The only thing that's changed is the mRNA sequence that's loaded into those little drops. And so all of the human trials testing we did, which was actually some of the most aggressive and extensive testing ever done for vaccine technology. I mean, the number of people tested worldwide was well in excess of 50,000 different individuals that they tested before they released those vaccines. That testing is still valid because they haven't really changed anything except the little mRNA sequence that's packaged inside of those lipid drops. So there's so little change, there's no justification for repeating all of that testing and delaying the benefit that we can get from releasing them quicker, right? Because if we wait and redo all these clinical trials, it's going to be at least 3 to 6 months delay. By that time, the next variant's going to be out and we're going to be behind the game.
Yeah, so the actual vaccine itself isn't really changing. It's just that like sequence that they're getting our body to react to that's changing.
Exactly. We're just updating it for the current variant that's out there and is dangerous to the population right now. And it needs to be timely. If we delay that at all, we don't get the benefit of the updated vaccine.
That makes sense. And as we're heading into this cold and flu season, and there's a new vaccine, a new booster out. The next question is that my pharmacy has sent me a notice asking me to schedule an appointment for both my flu shot and the COVID-19 booster at the same time. Is it safe to get them at the same time?
Is it safe? Absolutely. Is it recommended? I would. I mean, if you're going to take a day off or, schedule it so that you can recover over the weekend, get them done at the same time so you don't have to take 2 days off, right? Because if you get sick, or just ill feeling and don't feel well from the COVID, you're probably going to have that same reaction against the flu vaccine. So just double them up. We know that children get 3 or 4 vaccines at the same time when they're going through their childhood vaccine regimen. And it's completely safe. Your immune system can handle multiple pathogens at the same time. I mean, think about it. If you were to cut yourself when you're out playing, when kids are out playing in the dirt, right? They cut themselves all the time. There's not just one organism that gets in there. There's thousands of organisms that are in the dirt that you get exposed to when you cut yourself. They get in there and your immune system has to handle all of them at once. So it can certainly do the same thing with multiple vaccines at the same time.
Okay. So then building off that, I would assume then that it's safe for children to get the COVID-19 vaccine at the same time as they're receiving other childhood immunizations?
Yes, absolutely. Same reasons. Yeah.
Because your body.
I mean, we have the number of immune cells we have in our body is amazing in its arsenal. We have trillions of immune cells floating around in our body, and every single one of those B and T lymphocytes is unique to a particular pathogen protein, right? And so the array of organisms we can respond to at any given time is simply amazing.
Wow. Human body is an amazing thing.
It is. It's fascinating.
We have just about 5 minutes to go. So if anybody else has questions, I'd encourage you to go ahead and add those in because we just have time for a couple more. In the meantime, we have a question about immunity again here. So how come COVID immunity doesn't last for a long time, like the chickenpox immunity, for example?
So I talked about earlier in this that, COVID's kind of sloppy. That's how the different variants arise. And it's the new variants that are responsible for getting reinfected again. The chickenpox virus doesn't change over time and it doesn't leave you. So what's different is the chickenpox virus is in this family of viruses called herpes viridae family of viruses. They're lifelong infections. Once we acquire them, they never leave us. So if you get chickenpox as a child, that virus goes and it hides in your nerve cells your entire lifetime. We never get rid of it. And then when your immune system starts to wane and be less effective when you get older, it can reactivate. And when it reactivates, it comes back as shingles. So most of the time, shingles is not a new virus that you were exposed to. You had it hanging out in your body for 40 or 50 years. And it was just within your nerve cells, which your immune system is designed not to attack, right? It's kind of a privileged site. You don't attack your own nerve cells because that would cause permanent damage to your body. So because of this adaptation, the chickenpox virus just doesn't mutate much, hardly at all. So once you're exposed, you're protected for 40, 50 years until your immune system starts getting not so good.
So it just kind of hangs out there and then comes back. Now, so does that in turn mean that the COVID virus is leaving our body after we get sick in a different way than the chickenpox virus does then?
Yes. So most people really do rid themselves of the virus and it's undetectable after about 10 days following acquisition of the infection. The exception to that might be with long COVID, although we're not really sure. We can see persistence of the protein signature in particular cells in people who have long COVID, but we can't see very well or very often though is persistence of the nucleic acid, the actual virus production itself. And we're not sure why that is the case. And we're still really actively trying to learn more about exactly what long COVID is and why it's caused. But it doesn't appear that it's active virus production any longer.
Okay, interesting. Well, we have one more question about Omicron, and then I'll turn the floor over to you if you have anything else you'd like to add before we wrap up.
Sure.
Our last question here today is, I have heard that you're more likely to get the Omicron variant of COVID than you were previous variants. Is it true that the Omicron variant is more contagious than previous variants?
Yeah. And so that's why we have all these different variants of concern. The virus has made a significant change in its structure so that now your immune system is no longer as effective. And the Omicron seems to have changed the folding structure of that specific spike protein on its surface that it uses to dock to our cells in a way that actually makes it bind tighter, so that once a virus gets on the surface of the cell, it has a much higher likelihood of having a successful infection, causing a successful infection. So it's just, it's randomly evolved and it's better at doing its infection thing. So yeah, we are more likely if we are exposed to get a productive infection with the Omicron.
Makes sense. Well, we're going to slip in one more question that just popped up in chat before we wrap up. What is the current recommendation in regards to children returning to the classroom following COVID infections?
The CDC has backed off on the number, on the amount of time. So it should be 5 days after testing positive for COVID that children can return to the classroom, but they still should wear a mask for probably another 5 days after that. But their active production of virus has waned enough that with a mask, we're pretty safe from them infecting other people at that time. So it's 5 days after development of symptoms and testing positive.
So with that, we're at the end of our questions. Is there anything that we haven't covered that you'd like to make sure that we touch base on before we wrap up today?
Yeah, actually, there's one thing that I don't know whether I emphasized it enough during our earlier discussion about this new mRNA virus technology. The reason I'm really excited about it, and most immunologists worldwide are excited, is because it has really opened up such a new avenue of creating vaccines against things where we haven't been able to hit them before. So, for example, I pulled a review paper that focuses on all the different clinical trials being are ongoing right now to use this mRNA technology against other infectious diseases. And it's influenza, Epstein-Barr virus, HIV, cytomegalovirus, rabies virus, which we have not been able to really create a great vaccine against, Zika virus, Lassa fever virus. But the other part is it also, because it's such a platform kind of a technology where you can literally just put in a different mRNA and get a different result, target a different thing without dramatically changing the technology itself, it's also allowed us to now start developing personalized cancer vaccines. So we can literally take some cancerous tissue from a tumor, isolate out the mutated proteins, protein coding genes, I should say, from those tumor cells, put them into a vaccine and rev up a person's immune system so they can clear their own cancer without chemotherapy.
Wow.
So we're really, I think this mRNA technology is going to garner their inventors a Nobel Prize in medicine within the next decade, because it really is such a groundbreaking and exciting new invention in medical sciences. And I don't think that's been emphasized enough in the public realm.
Well, that is a fabulous note to end this on. Thanks.
Bye, everyone. Thanks for joining us.
Goodbye.